Pleasure to introduce Johanna. Johanna is originally from France. She did her master and PhD at the University of Lorraine, and I guess working with Marc Chassidon and also with Roberts. And at that time, she already started using ion probe SIMS for, at that time, oxygen and silica isotope analysis. And when she followed with it, with a postdoc at UCLA with using also SIMS techniques, mostly applying to meteorites and to ancient rocks. And she returned to France and did another postdoc at Institut de Globe, IGPG, in Paris, and when for 4 years had assistant professor position at university in France until she moved to Switzerland, where she is assistant professor now and director of SIMS facility. So I pass it to Johanna. Take it from here.
Thanks, Andre. It's a pleasure for me to talk today, and especially because I start this journey on interest on iron and sulfur biogeochemical cycle by a collaboration with Andre a few years ago by studying some pyrite nodule from the Barberton Greenstone Belt. So today I want to discuss about or to present you some results about pyrite, and especially about sulfur and iron isotope composition and the implication for the Archean biogeochemical cycle of iron and sulfur. So can I move? Yeah. So pyrite are a very interesting mineral. So it's iron sulfide that we can find everywhere. It's ubiquitous in all geological record, but we can also find them in any kind of different environmental setting, like in lake water or marine setting. Here, it's a picture of a confocal microscope picture that you can see some microorganisms and cyanobacteria, and this tiny green blue dot here are pyrite grains that are formed through the activity of these microorganisms. So pyrite can be formed through the activity of through the biological activity, and here is another example of rhomboidal pyrite. Sorry, but we can also have this beautiful cubic pyrite, and in the Archean record we have a lot of pyrite in mostly in shale. Here you can see this beautiful nodule. And we also have some pyrites that have been, that is detrital, as you can see here on this picture, some rounded grain. So we can find them everywhere, and it's very important because it can be formed through different pathways, quite complicated, that involve both sulfur and iron. And here is a schematic from Jorgensen a few years ago, where you can see the transformation between sulfate from the seawater through various tape and various sulfur intermediate, and iron 3+ that can also be reduced, and that end at the end with a pyrite burial. And so there is a strong link between the sulfur cycle, the iron cycle, and the carbon cycle, that we can study through pyrite investigation. So in the following, I will discuss mostly two pathways that are the most predominant to form pyrite. That is a monosulfide pathway that we form makinawite, that is this FeS precursor, then can be transformed in pyrite later. And there is a polysulfide pathway that is also include a monosulfide precursor, but mostly elemental different sulfur intermediate to produce pyrite. And so what is clear is that most of this reaction occur during diagenesis and here in the sediment. So it's pretty rare to form pyrite in the seawater or in a lake water column. But it's very easy to form in during diagenesis. And as I said, there is a strong, so this is another picture that is more a simplified diagram between the formation of pyrite and the carbon cycle. And so here you have the sulfate from the lake water of a marine setting and the organic matter pool. And you can see the coupling between the sulfate cycle that will be reduced in H2S, but could be also reoxidized, and the organic matter through the sulfurization of this organic matter and the production of pyrite. So by studying pyrite in the geological record, we have information about the iron and sulfur cycle, but also about the potential microbial activity that are important for reducing the sulfate species or the iron species, and also the oxidation in some case, and the carbon cycle. And what it has been proposed, but since quite long time now, is that in the Archean geological record, we have this nodule with a detrital pyrite grain. This is some beautiful example from the Witwatersrand Basin in South Africa that is well known for this occurrence of detrital pyrite. And the fact to have these pyrite grains that are round, and so that have been transported at the time of formation, is a strong indication of anoxia at that time. And so pyrite, in this particular case, is one of our good proxy for anoxic condition. And in most of the modern setting, also pyrite form in anoxic condition, but this is not true all time, and I will discuss that later. But the real smoking gun for the presence of anoxy during the Archean was the discoverance of mass-independent fractionation more than 20 years ago now by Farquhar and a collaborator on their pioneer work. And so sulfur has 4 stable isotopes that we are able to measure all of them since more than 10 years with a good precision. So you can see here the 32, the 33, 34, and 36 with their isotopic abundance. And a mass-independent fractionation is in a diagram like that, in a triple isotope diagram. So here we report the isotope ratio, the delta 33. So that is the isotope ratio of the 33 over the 32, and here the 34 under the 32. And so in a diagram like that, all rocks and every product we can measure on Earth will fall on this terrestrial fractionation line, with the slope is approximately 0.5, sorry. And what Farquhar and collaborator discover is that in the Archean, we have a deviation from this slope and from this line. And this deviation is what we call a mass-independent fractionation. And there is several notation to express them. So it's a S-MIF, and it's expressed as a capital 33 that you can see here. And so what is important is that you can have deviation above this line or below. And so you will have positive mass-independent fractionation, or you can have negative mass-independent fractionation. And what it was discovered at that time, and now have been investigated in a lot of detail, is that the Archean record have strong mass-independent fractionation. So this is one of the compilation from Ono et al. A few years ago, but there is a lot of different studies that have reference here. In the blue, you have the data that are produced by bulk experiments, so by ARMS data, so that by chemical extraction of the sulfide. And in white, you have the SIMS data by in situ, so the pyrite measure in situ. What you can see is that here we have this important mass-independent fractionation, especially here around 2.5. So that can be very large, up to 10 per mil, and that disappear completely around 2.4 billion years. And that have been proposed to reflect the presence of anoxic atmosphere during the Archean. And this disappearance around 2.4, sorry, is what we call the great oxygenation event. That is a major change in the Earth surface condition. And if, I think you all know this nice picture from Alliance et al., where you can see the variation of the PO2, either in logarithmic, in atmosphere, or in present actual level. So we are almost here. And you can see that during the Archean, we have no oxygen, or maybe some whiff of oxygen, or oxygen oasis. And then there is an increase of the oxygen level, but that is not so important during the Archean, or at the end of the Archean. And this oxygen level stay quite stable during most of the Proterozoic, and we have a second oxygen rise in the atmosphere at the end of the Proterozoic. And that is a major change in the Archean atmosphere, and that have strong implication for most of the biogeochemical cycle, but also for the microbial activity, because this oxygen is mostly produced by cyanobacteria or by oxygenic photosynthesis, and that implies the appearance and evolution of oxygen photosynthesis very early in the geological record. And so why the mass-independent fractionation is the smoking gun for this anoxia or anoxic atmosphere during the Archean? It's because the only way to produce a mass-independent fractionation of a sulfur species occurs through photochemical reaction and in an anoxic atmosphere. And so here it's also a schematic from Alevi et al. That express how you can produce mass-independent fractionation, here MIF in purple. And what you can see, is that you will produce negative mass-independent fractionation during photochemical reaction of sulfur species. And the sulfate aerosol will have a negative mass-independent fractionation. And so if the sulfate is there incorporated in pyrite, you will have a pyrite with a negative mass-independent fractionation. The other pathway is other photodissociation of sulfur species that can come to the elemental sulfur, and this elemental sulfur will have a positive mass-independent fractionation. And there have been a lot of work, I will not enter in detail, but there have been a lot of experimental work carried out first by Timmins, but by other group after that have characterized the effect and the influence of various chemical parameters, and also the magnitude of this mass-independent fractionation during photochemistry reaction. What is not clear still is how we transfer this mass-independent fractionation from this elemental sulfur or this sulfate through the geological record and through the Pyrite formation. And this is one of the questions that is interesting me. And so complementary to the fact that we have an anoxic atmosphere during the Archean, the sulfate concentration in the ocean was very low and was only a trace. And this is the recent, not so recent now, but a study of Crow et al. In Lake Matanao in Indonesia. And here you can see the record of the delta so this is the difference between the delta 34, so the sulfur isotope composition of sulfate versus sulfide from Archean pyrite. And so you can see that most of the delta 34 of the pyrite in the Archean are really close to 5 per mil, between 0 to 8 per mil. And this is the fractionation. So the difference, it's not exactly the fractionation, it's the difference between the sulfate composition and the sulfide composition, depending on the sulfate concentration in the water. And so you can see that with a very low sulfate concentration, we have a very low magnitude of this fractionation. And when the sulfate concentration increase, we will have a larger fractionation. And so in the modern seawater, when we have something like 60 micromole, depending of the environment, we can have up to 50 per mil or 70 per mil fractionation or difference between the sulfate and sulfide, when during the Archean it was lower. So this is most of the scientific consensus about what we think on the sulfur cycle in the Archean, even if there is a lot of unknown and debate in the detail. But I want to acknowledge that there is other view also for the sulfur cycle, and especially Omoto proposed that the atmosphere during the Archean was not so anoxic at all. They have found that there is other reaction like thermochemical sulfate reduction that can produce mass-independent fractionation, that is this thermosulfate reduction, even if this process that can occur at quite high temperature until 100 or 200 degrees, so during diagenesis or early metamorphisms. But this reaction produces a very small magnitude of mass-independent fractionation. But there is still a recent paper from last year from Omoto that proposed a seawater sulfate origin for early Earth volcanic sulfur, where you can still produce some mass-independent fractionation, but this is where in this article only in the stratosphere, so not in the troposphere. And then you can have some mass-independent fractionation that are produced by thermochemical reaction. And in this paper, he proposed to have quite a large amount of sulfate in the ocean. And so one of the aspects is that it's not so black and white as always, and so there was some oxygen oasis during the Archean. This one was a model paper by Stefan Lalonde and Kurt Conoser that proposed to have some oxygen oasis that was either in the marine setting, but also that was what they proposed in this paper, also on terrestrial land. And that you can have mat in a river or in lake. You can also have this desert crust in some arid setting. And all of this microbial activity can produce oxygen if there was some oxygen photosynthesis. And then you can have oxygen produced locally, and that will have been consumed locally, even if most of the environment was anoxic. And so the atmosphere was mostly anoxic for producing the mass-independent fractionation of sulfur, and the water column could be have been mostly anoxic with locally some oxygen oasis. And so I will not enter in detail, but I will be happy to discuss that later if you want. But there is a lot of different studies that propose oxygenated ocean before the Great Oxygenation event, and especially with recent advance in non-traditional stable isotope like chromium, iron, and molybdenum, and also thallium. So this is one of the paper. So I show you two quite recent paper. One on the Mount Microscale in Australia, where Ostrander et al. have combined molybdenum isotope and thallium isotope. And so they propose in this case that in this shale, we have some shallow waters that were fully oxic to produce manganese oxide that could have been later reduced during this in a more oxenic condition. And the other study is from the Nusne Group in South Africa, and it's even older, it's 2.9 billion years. And here the Ekman et al. Have measured the iron and sulfur isotope composition of various pyrite, sedimentary pyrite that you can see here in this white and black. And they propose that this strong variation in both sulfur and iron could explain only by the presence of oxygenated shallow water in the seawater. So there is some oxygen oasis, and that is complicated for the mass-independent fractionation record we have. Because if we look at this record in detail, and a lot of people have tried to explain the magnitude and the variation through the Archean. And this is quite complicated because first, we don't have a good stratigraphic record. We have some areas that we don't have any kind of rocks that we can measure. We also have some geological records that we don't have pyrite inside, so it's also more difficult to measure sulfur if there is no pyrite in the sulfur. Maybe with the advance of carbonate-associated sulfate, it will be we will have more information on that part. But what is interesting is that you can see that for one formation we have a strong variation. And we have a large range of variation in CAP 33 also, with a very few negative, but more likely a very positive, especially in this study, but in so other study. And we have a large range of mass-independent fractionation at a microscale. That is difficult to explain by only reflecting photochemical reaction in the atmosphere. So there is some other process during diagenesis and during pyrite formation that are going. And that is why a lot of SIM study have tried to unravel. And so here it's a study from Meyer et al. So it's in a 2.6 formation, Locamona Formation in South Africa. It's a neo-Archean microbial mat. And what they have done, so there is a lot of pyrite, as you can see here on the SEM picture. And they have measured this pyrite by a secondary ion mass spectrometry. So you can really measure each pyrite grain, most likely. What they found is that you have pyrites that are completely that are associated, that have completely different mass-independent fractionation, with this large, massive pyrite that is mostly positive, and this pyrite that is more porous and grainy, that is likely negative. And so it's clear that these two pyrites in the same sediment are not reflecting a different or a variation in the photochemistry of the atmosphere, but more likely different process of pyrite formation in the sediment. And that have been also proposed a few years ago by Desiree Rording during her PhD. So she has measured a lot of pyrite and sulfide from the Barberton Greenstone Belt. She also have measured a lot of barite. And what she shown a very large range of sulfur isotope heterogeneity. And she proposed a quite complex model that you can see here that can explain this heterogeneity with various sources of sulfur, so that you have sulfate from the seawater, you have elemental sulfur that carry some mass-independent fractionation, and you have juminal sulfur and magmatic sulfur that is also incorporated. And you can also have some biogenic sulfide from the reduction of sulfate from the seawater. So quite a complex way of formation of pyrite. And this is also what we have shown a few years ago in a collaboration with André and Axel Hoffman. So this is from the Buck Greenstone Belt. It's a 2.7 nodule. So they are beautiful centimetric pyrite nodules that have been measured before by bulk, so by chemical extraction. And the bulk value of this mass-independent fractionation of this nodule was 0. But when we have performed some SIMS analysis, you can see here, this is a bulk value that is close to 0, and this is a CAP 33, and you can see that we have a huge range of variation, almost 5 per mil, from the core to the rim. And the core and the rim are different mineralogically. We have a more porous core and a more well recrystallized rim, and we have some trace elements like nickel and cobalt that are more enriched in the rim that show a recrystallization. So when we study a nodule, we know that this nodule record diagenetic process, but still during this diagenesis process, we also have another step of pyrite formation with late fluid that recrystallize part of this pyrite nodule. And it's more and more suggests that pyrite do not record global environmental conditions that have been proposed by David Fyke a few years ago. And there is a recent study by Pasquier et al. This year that have measured pyrite from quite modern pyrite compared to Archean from the Mediterranean Sea. And what they show in this paper, this is the difference between the sulfate and the sulfate because they have the sulfate composition. And what they show in this paper is that the sea level that is here control at least partially the difference between the pyrite and the sulfide. So this pyrite is not recording a global change in the environment, but more some local control during the pyrite formation. And it also have been shown by Pace et al. In some the 1.9 formations from the Naigo, from the far deep drill core, where they have measured pyrite this time. So this last study was done by bulk analysis, most likely. But in this study, and this you can see here this beautiful schematic. So they have measured the pyrite here, but they propose a very complex model to explain the sulfur composition of pyrite that implied two different microbial metabolisms: the microbial sulfate reduction and sulfide oxidation, and some variation in the amount of sulfate and the sulfate diffusion in this quite restricted basin. So, and the conclusion of this study is also that pyrite is not recording any global change, but a local environmental control of the basin at that time. So all of this study tell us to be careful when we want to draw a big picture from a very careful study from the Archean. And so this is what I want to show you, three different studies on Archean pyrite, and where are the caveats and where are the promises. So first, I want to show you some recent data from the Buck Reef charts. So this is a beautiful charts formation in the Barberton Greenstone Belt. You have here the map of the Barberton Greenstone Belt with these three formation or three groups: the Onverwacht Group, the Fig Tree Group, and the Moody's Group. That is going from 3.5 to 3.2 billion years. And these Buck Reef charts have been drilled by ICDP a few years ago, and I will show you some result of the Barb 3 drill core. And so here, this is a picture of the chart in the field. So a chart is it's a siliceous sedimentary rocks that have been deposited in a marine setting. So the Buck Reef charts have been heavily studied from quite some decade now. So it's almost it's a 800 meter of charts with carbonate that are deposited in various environment from deep water to shallow water and even evaporitic setting. And here you can see the stratigraphic section from Theiss, and with a relative sea level reconstructed. Sorry. So there is a strong variation in the sea level during this Buck Reef chart deposition, and this is the last log drawn from the ICDP Barb 3 drill core from the PhD project of Alice Pellan in biogeoscience in Dijon. The Buck Reef has been heavily studied because it's one of these formations that record a fossil microbial mat. And this fossil microbial mat could have been interpreted as possibly photosynthetic. So there was several studies by Tyson Law and ties after that, that shows this microbial mat that you can see here. There is some organic matter. This Buck Reef chert is very enriched in organic matter. It's for most of it is a black chert with various forms of organic matter. We have some clot, but we also have this film. And one recent study of Johanna Carbonne et al., who has characterized the structure, the chemical structure of this organic matter. So it's not exactly the same film than in Tice and Low, but when you have one of these black cherts from the Buck Reef, you can see that there is a lot of different organic matter film here on this SEM picture, and you can see this the carbon picture and the silicon picture. And he has studied them by Raman and by synchrotron technique at the carbon age. And what is interesting is that even if these Buck Reef cherts have been quite metamorphosed, because the Raman temperature is around 350 degrees, you can still see that we have preserved a large heterogeneity in this film. So this is all of this film on, so they are very close together. It's in the same chert sample, and they all have different chemical structure of the organic. So what is the origin of this heterogeneity is not completely clear, but could be completely consistent with a biological origin, but also with some aerosol organic particle like on Titan. So there is still some work going on to better understand the origin of this organic matter and the film. But it's quite interesting. And so what has been done, and it's have been done in a collaboration with a lot of persons who has worked on this Barb3 drill core. They have measured the sulfur isotope composition in bulk in pyrite by SIMS at the North SIMS, that was during the PhD project of Alexandra Galish, and in siderite, but there is only very few data points for siderite for iron isotope, and I will now discuss iron isotope on that. So this is the drill core of the Barberton with a dip. So this is the dip, sorry, and this is the delta 34, the cap 33, and this is the recent data we have done on the Swiss SIMS. So most of it are consistent, but we have target at the Swiss SIMS more smaller pyrite than in previous study. So we can see that there is variation through the drill core of both of the delta 34 and on the cap 33, with some areas that have recorded quite strong mass independent fractionation. And so what is the control of the sulfur isotope composition in pyrite? Is it environmental? And so we can really see a difference between a deep water and a shallow water environment, or is more a diagenetic control during the formation of pyrite. So first, if we look the pyrite morphology and pyrite diversity, we have a lot of different pyrites. So this is this kind of film that are possibly microbial mat, and you can see it's there is some organic, but it's most likely chlorite, and there is this small pyrite grain. We have also what we call disseminated pyrite, so that can be very disseminated or almost aggregate, and we have huge pyrite nodule, and there is no very strong control of the stratigraphy on this pyrite formation. We can find pyrite nodule at various places, and we can find disseminated and lamina at various places of the drill core too. We have done some trace element in this pyrite, and what is interesting is that there is no difference between the pyrite in the disseminated and the lamina. We haven't run the nodule pyrite right now, and all the cobalt nickel ratio are below 1, and so pinpoint to a sedimentary origin of pyrite. But what is maybe interesting to dig a little bit more is that we have a slight enrichment in selenium and arsenic in pyrite associated with the lamina. So what I call the lamina is this kind of structure that can be a microbial mat. And so here I show you the result we have in a triple isotope diagram. So this is a cap 33, the delta 34. And the color is from the different type of pyrites. So the pyrite in the lamina are the ones that have the strongest mass-independent fractionation. So they are very tiny pyrite grain. For the massive and nodule pyrite, so we have some very large pyrite grain that is not really a nodule, that is larger than the core, basically, so that we call massive. And you can see we have two different signature in mass-independent fractionation. We have some massive pyrites that have mass-independent fractionation and nodules that have less independent fractionation. And the disseminated pyrites show the larger range of variation in delta 34, but very low mass-independent fractionation. So first, what is interesting is that this diamond square here are the bulk isotope data for these three samples. And so you can see that we are missing some information, especially in the lamina, because we, in this study, for now, we have analyzed only the lamina and not the pyrite in the matrix. So with the bulk result and this discrepancy, we can estimate that the pyrite in the matrix are more, have less mass-independent fractionation than the pyrite in the lamina. So we have different pyrite generation, and even we have pyrites that are early diagenetic, and we have pyrites that are late stage crystallization, like this nodule with juvenile sulfur incorporated. We don't find a very strong record of sulfate reduction in the lamina, but it's still possible, but not so clear for the disseminated pyrite. What is interesting is that the pyrite in the lamina could have been formed through the organic sulfur as proposed by this recent study of Facret and Castev, and we will still continue to investigate this hypothesis. But what I want to show you here is that this sample, or all of these samples have been hit at 350 degrees. They have all record fluid circulation. There have been a debate on the influx of endothermal fluid during the Buck Reef chert silicification. And what is clear is that not all of the pyrite have been formed during this late stage with juvenile sulfur. We still have signatures that are preserved. But it will be difficult to interpret and reconstruct a global sulfur cycle from the bulk data for sure, but also from the SIMS data at that point. So now I want to show you another study from another chart from the same Barberton Greenstone Belt, that is the Marin Formation. So you still have this map of the Barberton Greenstone Belt. And this time we are not in the Onverwacht Group, but we are in the Onverwacht Group, but we are at the top of the Onverwacht Group, just before the transition with the fig tree. And this formation have been drilled by IPGP a few years ago, and this is the drill core that have been recovered. So there is also part of the fig tree and the Mapepe formation that have been drilled during this drill core, but today I will show you only results from the Marin formation. So the Marin is also a chert, and that have been deposited just after comatite. But this comatite are heavily silicified, and they have recorded a lot of fluid circulation. And the Marin charts record also a lot of fluid circulation. But the Marin charts have been also heavily studied. And there is a very nice study by Troer and Lowe a few years ago that have reconstructed the sedimentary environment. And so we can estimate that the charts have been precipitated or deposited in a very quiet water setting, in quite deep water. And so the formation of charts could be discussed. So it's not a chemical precipitated, but maybe imply silica granule formation. It's true that we have this granular chert with a lot of this silica particle inside, but it's a chert that have been deposited in quite deep water and in a quiet setting. There is some stromatolites that have been described in the Mandron chert formation. So here you can see this a picture from the field and a picture here in scene section. And there is a lot of this also lamina and organic matter enrichment, but I will not discuss that so much. What we found when we opened the drill core is that we have a lot of this fluid circulation. So it's really rich in pyrite. You can still see them with a naked eye. But we have a lot of this fluid circulation. You can still see that we have the lamina and we have some sedimentary structures that are preserved. We have a zoo of pyrite. We have very small grain. We have pyrite in the fluid circulation. We have huge cubic pyrite that you can see. We have aggregate, and we have very small pyrite grain. And also there is no real stratigraphic control of the pyrite morphology and their location in the drill core. So we have reconstructed the sedimentary environment of this particular drill core with the help of Nadia Drabon. And we have measured the iron and multiple sulfur isotope with the SIMS. So here the star is a sample, and this is the depth of the drill core. You can see we have a huge variation in the iron isotope composition, because this is almost the entire range of terrestrial value between -4 to +4. So we have that in one drill core from chert in the Barberton Formation. And we have also large variation in sulfur in delta 34 and in cap 33. Even if you look, we have most of our samples that have very small magnitude of mass independent fractionation, and 2 and almost 3 samples have mass independent fractionation. There is no control of the sedimentary environment on this composition. And so we tried to understand what was the record of this sulfur and iron isotope composition. This is also a triple isotope diagram with the mass independent fractionation and the delta 34. And this is the sample with their depth. And you can see we have some mass independent fractionation, but not in all samples. And most of the samples have a close to 0 mass independent fractionation. So we look back to the mineralogy of the pyrite. First at all, they are all diagenetic. They all have inclusion of cherts or quartz, or even some organic matter, even some of the small one. And so what we have done is that we have done some focus ion beam scene section, and we observe them at the electronic microscope. And what was a surprise is that some of these aggregate pyrite that look completely recrystallized, if you look them at the atomic scale, they are completely polycrystalline. So this big pyrite is composed of a lot of small nanocrystal of pyrite, and you can see that they have different orientation, as you can see here with this different pattern. And we can also see that on the diffraction pattern here. And this cubic pyrite is more likely what you expect, is that it's a recrystallized monocrystal, and you can see here there is only one pyrite crystal that compose this entire pyrite grain. But what was interesting is that this is only the polycrystal pyrite that have the mass independent fractionation signature, where most of the monocrystal, they don't have mass independent fractionation, and they have close to 0 cap 33. So that was interesting, because this is the same. The Barberton Formation have been hit as almost all the Barberton Greenstone Belt, around 350 degrees. And so it have been recrystallized, but we still have pyrite that are composed of this poly and small crystal grain that still have quite high mass-independent fractionation. And so when we saw that, we can ask different questions. So maybe we have different pyrite generation. So 3 different pyrites that have been formed at 3 different times. But we don't have a very good answer for that because there is no correlation between either the size or the morphology and the isotope composition. There is no correlation with the stratigraphy. We also have done trace element, and there is no, even in the large cubic pyrite, there is no detectable nickel and cobalt or zinc enrichment. And all the nickel cobalt ratio are below 0.6 for all the pyrite group. So they all have a thin sedimentary origin, and they not have been formed during this late stage fluid circulation. And also because most of these pyrites are very big, like 100 of micron, we were able to perform some isotope profile. So for iron and sulfur, but here I show only the cap 33. And in all 3 groups of pyrite, when we do that, we still find some mass-independent fractionation at the core of this pyrite. As you can see here, most of the pyrite have a close to 0 mass-independent fractionation, the same here. But we have one, in some 2 points that have a mass-independent fractionation in the core. And for the polycrystalline pyrite, there is no difference, and so there is a very homogeneous mass-independent fractionation through the profile. So we try to explain the iron isotope composition of this pyrite, because as I showed you before, it's a very large range of iron isotope composition. And there is this study that have been proposed a few years ago, but have been also there is several more recent study, especially by Andy Hurd, that also suggest this hypothesis that the iron isotope composition of pyrite is not also controlling by any environmental or biological activity, but just reflect a difference between a kinetic and an equilibrium effect. And so more you precipitated a pyrite at equilibrium, the pyrite will have more likely a positive delta 56 value. And if you precipitate a pyrite in a more kinetic regime, you will have more a negative value. And so this pyritization rate or pyrite formation rate can explain why we have a large range of pyrite formation. So that have been proposed for our pyrite grain, but we should have reverse profile, sorry. So maybe for this one, it could be possible, because you will explain to have more kinetic effect and more isotope equilibrium effect during after recrystallization. So it's still possible, but it's not so conclusive, I will say, in the short study, but I will be happy to discuss. What surprised us is that if we draw the probability density of the iron isotope composition of this pyrite, you can see that we have a bimodal distribution, and the difference between the two is almost 3 per mil. And this corresponds to the fractionation expected for dissimilatory iron reduction. So this is a microbial metabolisms that can reduce iron oxide. So here it's a schematic from Crosby et al. You can see a cell or microbes that will reduce the iron 3 and the reduction and produce iron 2 that can be later incorporated in pyrite, and the fractionation will be around 3 per mil. So we propose that we have this dissimilatory iron reduction recorded in Mandon, and that we have a partial iron reduction in group A that is completely consistent by the presence of iron oxide. We still have some iron oxide in this sample who has negative iron isotope composition, when we don't have any iron oxide in the sample who have potential who have positive iron reduction. And this could be also consistent with the polycrystalline nature of the grain for the positive pyrite. And so we propose that we have the dissimilatory iron reduction as 3.26 billion years. So it's not completely new. There is a lot of phylogenetic studies and laboratory culture that suggest a very early onset of iron reducer. There is geological evidence at 2.7 and 2.5, and also suggested at 3.8. And so we propose kind of a complex model when we still have an anoxic condition, but how pyrite are completely formed during in the sediment, and that you have this reduction. So this dissimilatory iron reduction is occurring in the sediment and not in the water column. And later, due to fluid circulation, we are resetting the sulfur isotope composition, but not the iron isotope composition in this case. And so maybe iron isotope is a little bit more resistant than sulfur for metamorphisms, but that needs to be confirmed yet. And so what in this case, pyrite record most likely some local process of formation in the sediment and not at all a global signal of the water column. So we cannot really conclude on either the presence of oxygen or not in the water column during the time of the deposition of the Mandon charts. And so the last study I want to show you is from the Tumiana Formation. So this one is 2.7 billion years in the Pilbara Craton. So we are going to Australia. It's also have been drilled and it's deposited most likely in a lacustrine settings. So the Tumiana Formation is very well known because it's incredibly well preserved stromatolite. This is a picture from the field, and this is a thin section. What is incredible is that you can see this beautiful dome in a field, but when you do the thin section, you can still see all of these small stromatolitic dome that are growing or that have been preserved in this formation. So it has been studied for more than 20 years now, and there is a lot of consistent signature of trace of life in this Tumbiana Formation. First, the morphology and the sedimentary environment are completely consistent with an ecosystem, but there have been some isotopic evidence of metanotrophy, acetyl-CoA pathway, nitrogen fixation, also some possible iron metabolisms. And these have been also studied because the Tumiana Formation record what we call the Fortescue excursion, that is this very large excursion in carbon isotope that should have reflect metanotroph metabolisms at that time. But what was not so well understood is that if there was some sulfate reduction, and so there is also the question of anoxic and oxygenic photosynthesis, but I will not go in detail for that. But I'm more interested about if sulfate reduction were present at that time, because in modern stromatolite, sulfate reduction is a key component for carbonate precipitation. And so it has been studied during the PhD of Christophe Tomaso by Bulk, but they didn't find a very consistent signature of sulfate reduction in the bulk sediment. But what was observed is that we have a lot of this micropyrite in Tumiana stromatolite. So this is some beautiful synchrotron picture from the PhD project of Marie-Catherine that have been published also. And so here you can see the map of the carbonate and the map of the manganese, and you can see here the map on the iron, and you can see that the lamina here that are enriched in organic matter are full of pyrite, and these pyrite are very small, they are less than 1 micron. And they are completely associated with the organic matter. This is a Raman map where you can see the organic matter in red, the carbonate in blue, and this tiny blue dot here are all the pyrite grain. So we have developed with Laurent Remusa, a new protocol to measure the sulfur isotope by the nanosims, because these pyrites are too small for the conventional sims, because they are less than 1 micron. So what we have done is that we develop a new protocol to measure them by isotope imaging. And so for that, we have done by the nanosim. So we can acquire some very nice picture and nice isotope image of the carbon, but also of the carbon and nitrogen here, but also on the sulfur. And we can measure the individual, or we can extract the individual sulfur isotope composition of this pyrite grain. Because we are measuring very small grain, the precision we have is very poor. It's around 2 to 3 per mil, depending on the session. But for large range of variation, it's already giving us some information. And what we found in this Tumbiana formation is that this micropyrite have a very large range of sulfur isotope composition with values that are down to -33. So quite very low and completely consistent with the microbial sulfate reduction origin for this micropyrite. What is interesting is that if we measure the bulk value of this sample, because we have a lot of this pyrite grain inside the carbonate, the bulk value give you a typical Archean sulfide value around +3 or +4 per mil. So it's only this micropyrite, and they are not the main pyrite component in this stromatolite that have this very small and very low value of delta 34. And what is interesting is that the size of this pyrite is also completely consistent with the microbial origin because they are very small and quite polycrystalline. And so I want to show you also that has been also analyzed in Strelepool, so in another emblematic stromatolite from the Archean. This is a 3.4 billion years. And in his study, Tommaso Bontonieri have measured most the organic sulfur, so that was already quite an analytical achievement. But in the same study, he has measured 9 micropyrite that are also very small. But in this case of this Strelepool, they are not associated with the organic lamina. They are more likely disseminated in the carbonate. But these 9 micropyrite have values that are down to -18 per mil, also consistent with the microbial sulfate reduction, especially in a low sulfate environment. And so maybe these micropyrite are a very good proxy of biosignature of microbial metabolism. So it will be interesting to measure both the iron isotope. So in the Tumiana Formation, this is a current PhD project of Marin that have measured that. And on Strelepool, they are very small and quite difficult to already observe, but this will be a going on project. And so what I want to say is that if you have some beautiful stromatolite that you think there is some micropyrite inside, we will be very happy to investigate them for continuing this study, and also for mineralogy, because I really think that this micropyrite could be a very good proxy and a very good biosignature of microbial activity in the Archean. So in conclusion, I think I hope to have convinced you that micropyrite can record microbial sulfate reduction and in stromatolite, and there is still some ongoing work for the iron metabolic activity. What is clear is that for each Archean sediment we are investigated, we have each time multiple origin and multiple generation of pyrite. And it looks like the sulfur isotope composition is more likely reset by fluid circulation, by late fluid circulation, than the iron isotope. But that needs, we only have done 2 studies on that, so that need to be confirmed. And I think that in situ isotope analysis associated with detailed mineralogy is a key to better understand the iron and sulfur biogeochemical cycle since 3.4 and 3.2. Because at that time, if we just draw a big conclusion from bulk analysis from sediments that have been metamorphosed at 300 degrees, we are making mistake because we are also integrating some late stage pyrites that are just recording late stage fluid circulation. And I think I want to thank, this is not studies that I conclude by myself. I have a huge team of nice collaborators since many years that I want to all thanks, and I also want to thank the funding agency. And I think I'm happy to take questions.
Great, thank you, Johanna. We're going to have to give people, we always have to give people some time to put their question together, but we are open for the discussion session.
So maybe, John, I start as other people come with questions. So I have two questions. One for iron isotopes.
Yes.
You showed that you suggest iron reduction starting from, I guess, as early as 3.8. I'm just curious if you have any thoughts on a process that led to iron oxidation. Obviously, you need to oxidize iron.
Yes.
And the second question is on sulfur isotopes. And you know, in early days, when people sort of calibrated at what level of sulfate you get large fractionation. At that time, large sample size was used. And now with elemental analyzer and iron probe, we go into smaller and smaller samples, and we find much larger negative fractionation, which we're trying to interpret using the same sort of approach that it indicate either higher sulfate level in local environment or biological sulfate reduction. So I'm wondering if you have any thoughts, like how do we reconcile this new information from small scale studies to this fundamental approach, or which based on much larger samples? And specifically, how common is this highly negative sulfur isotope values in your micropyrite? Is it just a small fraction?
Okay, so maybe I start with this second question. The problem is it's quite, I don't know. It's always difficult when you measure that at this scale, but there is quite a. It's not one point when we measure a very negative value. Even in Tommaso study, there is quite a few pyrites that are very negative delta 34 value. But there is only 9 micropyrite grain. In our study, I think we have measured something like 100, and I can show you the distribution, but we have some quite a large number that are below -20 per mil variation. So I think these micropyrites are negatives, and I think this is what we need to do in the future, is that to measure more and more of them, of this very small micropyrite, and so that we can see if there is a pattern, and that if we can use them to reconstruct any kind of sulfate level. But for now, we don't have we have this only 2 studies that are really measure this very small pyrite, so we cannot conclude on that, and we cannot extrapolate from these 2 studies neither. What is interesting is that to reconcile what we know for the sulfate level, I think it's because that was the same is that more of the modern or the study in modern environment are done by bulk, and so we should start also to investigate this modern environment by in situ and by measuring this pyrite in this lake or in modern stromatolite and even in marine setting. And so then we can also compare, because we should also have negative, very negative pyrite in this environment, even lower than the bulk. So I think this is one of the possibility. And also in the Strele pool and Tumbiana, I think even if the sulfate concentration of the environment was low, you can still imagine that if we are in microbial mat, you can concentrate somewhere this sulfate level, and so at the scale of the microbial mat, you have enough sulfate to express the fractionation. This is also one possibility, but in this case, that really means that pyrite record only very local process during that formation and not something global. This is not because you measure very negative pyrite in Strele pool that you have a lot of sulfate in the Strele pool environment, but maybe locally in the growing stromatolite, you will have enough sulfate to have these microbes that were happy and fractionate. And for the second question, I think this is true that if we, there is this big question about what was oxidant for iron during the Archean. I saw several studies recently that proposed that anoxygenic photosynthesis was more common than thinking. I think there is a lot of groups that are suggesting that the presence of this anoxygenic photosynthesis metabolisms very early in the Archean, so that could be a possibility too. But this is also something that should be more investigated in detail, for sure.
Okay, great. Greg Vitalik also had a question. I see that you're already unmuted. Would you like to go ahead?
Yeah, sure. I'm amazed. You've emphasized. It's a really great talk. You've emphasized stromatolites. But what you're finding is so different from what I find in the rocks that I'm looking at in the Archean. What I find in my rocks, which are evaporites, which I regard as, you could call them playa lakes if you like. I regard them as paleosols because playa lakes are dry most of the time. I'm getting loads of framboids, and yet you didn't show a single framboid in any of this stuff. And yet you have really clear evidence of biological fixation. These bugs are not making framboids. We seem to be looking at different worlds here.
That is really interesting. So first, I don't have for Strele pool. I have only one sample that is the same than Tommaso Bontoniali, who showed me. So I don't have a complete picture of what he was, but I was in contact with Abigail Wolf that didn't because I was asking for more sample, and she told me that it was not so much sulfite. So I will be happy to see if there is some framboid. I never saw any framboidal in any of the charts or the formation I've found. But maybe this is a question of sample. Maybe I don't have the same exact sample.
I think it's a different environment because mine have a lot of sulfate evaporite pseudomorphs in them.
Okay.
And the framboids seem to be in that. Could I send you some samples?
Sure, I will be happy to look for this framboidal.
To do with as you will. Yes.
Yeah.
It seems.
That will, yeah, no, I will be happy to look for any kind of Archean framboidal pyrite. I think it would be very interesting.
So I'd always assumed that microbes made framboids, but some microbes don't, evidently. That's what you've shown us. In your environments, in your stromatolitic environments, they're not making framboids. That's odd to me.
Yeah.
Do we have modern experiments that show that microbes making just funny looking little crystals?
There is no, I don't know. Yeah, maybe there is some experiment going on that. But we have measured rhomboidal pyrite in different environments, so that we can, and there is also micropyrite in the same environment, and they are not recording the exact same condition. So that could be one possibility too.
Excellent, thank you.
Okay, at the moment, we don't have any more questions in the chat, but by all means, we can continue.
Yeah, maybe, Johanna, I ask you more on sulfur.
Yes.
So if you would average your micropyrite, like, I mean, it's hard to do it, to go from ion probe to sort of bulk, but if you would try to give average value, what do you think it would be for your micropyrite? And second, like, do you think this highly negative values in presence of very low sulfate, do they imply very slow rate of sulfate reduction, but even on microscale you can produce this highly fractionated values? So what do you think is responsible for it?
Yes, sure. So it depends. For the micropyrite, we have more negative values. So in the case of Tumbiana, we don't have the same. If we average everything, we don't have the same values than the bulk value. But that is explainable by the fact that we have this pyrite in the matrix that are more large and that have more positive delta 34 value. And also, micropyrite is not the main. It's not the main component of sulfide. I think when you extract them, you extract preferentially, or you have more pyrite from the matrix. This is the same case for the Buck Reef. We have a completely different value from the lamina and from the rock sample. So we have a total discrepancy, but it's because in the matrix we also have this big pyrite. So I think the average of micropyrite in the few studies I've conducted are smaller or lower than the bulk value or than the values that have been recorded for these rocks. So that is for the first point. And then, yeah, I think this is completely possible that, yes, we have a low sulfate or cell-specific sulfate rate at that for expressing this fractionation. And what could be interesting, and this is one project we are still conducting with Julien Alléon, who is a specialist of organic matter, is that maybe this is due to the structure or to the nature of the organic donor in this case. And so we want to measure the organic matter that is associated to this micropyrite to see if there is difference from this organic matter that is really associated to the micropyrite, to the organic matter that is more in a film or in some other record. And so maybe the nature of the organic source was different, or for sure was different during the Archean than now when we look for any kind of experimental. But I think it's a very interesting field to dig, and there is a lot of experiment we should still perform in a lab to better understand how we fractionate. So for now, for example, there is no study that I'm aware of that have really produced this micropyrite in a lab and measure them and isotopically. I know that there is some mineralogy studies that have been done, but I never saw any in situ data on this pyrite produced by microbial sulfate reduction in any kind of experimental settings. So that would be also interesting to look for that.
Yeah, you potentially have very carbonates and stromatolites probably have very little organic carbon. So yeah, you can have relatively a slow rate of sulfate reduction. And even under low sulfate, you can diffuse enough sulfate, but you don't develop a limitation in the system.
Yes. But we need to investigate more stromatolite, I think, to have a better view. of the link between this micropyrite and microbial sulfate reduction and carbonate precipitation, for sure.
Yeah. And thrombolites are really rare in Precambrian, and I mean not just rounded shape, but sort of rounded with having internal small rounded shape inside. So they're, for whatever reason, very rare in Precambrian rocks. Just a comment for Greg. Okay, thank you, Johanna.
Thanks.
All right, do we have any questions or comments or points of discussion from anybody else?
Can I
Sure,
come
yeah.
into it?
Go ahead.
So maybe sort of a half question and half comment. So Joanne, you have nicely shown that in each rock you have investigated from the Archean, there are several generations and several origins and several shapes of pyrite, and they all show different isotopic signatures, which is something that actually could be expected. But could you qualify to which of them would you give the best or the most significance for the questions we're asking about being syngenetic and telling us something about life and chemistry? And I would say from what you have shown there, all of them are marine. And what Greg has said, he claims to have something from terrestrial environments, paleosols, which I find difficult, having sulfates and evaporites at the same time there. But could you give us a qualification? What do you think is the best signature and where should one look for? You had a beautiful example of laminated parallel to the microbial met pyrite concentration. Is that the best one?
I think, yeah, I don't know, because the study of Mandon was quite surprising, because we have this pyrite aggregate that we target, and first we were really thinking that, okay, this pyrite is late stage. And at the end, that was the one who has a mass independent fractionation and is still polycrystalline. And that was perturbing at the beginning. And we have done and redone the analysis several times, to really be sure. So I think we still have potential to have pyrites that do not have very nice morphology. We have a lot of this pyrite aggregate, or how we call it in the Archean, that don't have a very nice shape. And this can be a recrystallize of smaller pyrites. So I will still try to analyze them. But yes, I will go for the smaller one and the ones that are the most, if there is organic matter, and in this black shell there is a lot of organic matter, I will look for the pyrite associated with the organic matter in the first step to really look for them. But I think this is one of the problem and the limitation of the SIMS is that because we don't have a good morphological criteria, we need to do extensive study and to perform enough analysis to have a good statistical data on what is the pyrite on that.
Can I put two following questions? One is, you have also shown that even the very fine pyrite grains within the stromatolitic or microbial lamina still have different signature than the lamina themselves, the organic matter itself. So there is a difference. How to explain that? And the second question would be, what is, I don't know, I just don't know much about pyrite. I don't know, what is the recrystallization temperature of pyrite?
I think it could be very, very small. Any kind of diagenetic temperature you can recrystallize if you have the good condition. It's a good redox condition. And I think that is why the sulfur, and because most of the fluid are more enriched in sulfur, you will modified quite lightly the sulfur isotope composition. So that is why we have this part of the range of mass-independent fractionation we observe is just due to this, the fact that we have some recrystallized pyrite in the geological records that have lost their mass-independent fractionation during recrystallization with a hydrothermal source. And for the lamina, this is a good point. I haven't done so much of this work. I know that there is some groups that are really putting some effort to measure the metal composition and concentration in the organic, and that will be interesting also to have the concentration of metal in the organic and the concentration of metal in the associated pyrite. They will have been different for sure, because you will, if these pyrite are formed from this organic, they should not have the same signature from this organic, but that could be maybe interesting to look for it. For now, I'm just looking for the trace metal to quite be sure that they are not metamorphic from origin or very late stage recrystallization, that they still have a sedimentary origin, I would say.
So would you agree that we are basically looking at pyrite? We in most cases probably look basically at the whole history of the rock and not only on the early diagenesis?
Yes, for some of them for sure, and I think there is very few possibility of dating, and we try. So there is some groups that are doing radium osmium. I think that will be interesting because yes, I'm sure we will have some surprise if we are able to date them. Some of them will be quite young compared to the age rocks, I would say.
Thank you. I would love to send you some microbial met examples, not pyrite.
Okay, sure. I will be happy to look at it. No problem.
Okay. Well, if we don't have more questions, then maybe it's a good place to cut it. So thank you, everybody, and thank you, Johanna, for that today. It was really good.
Thanks a lot.
Yeah, and it seems you've got a couple of sets of samples coming your way, so that was a success, right?
Exactly.
All right. Thanks, everybody. We will see you next week.
Bye. Thanks a lot.
Thank you. Bye.